Welcome back to Introduction to Neuroscience, where we're going to get started at the beginning. This is the ABCs of neuroscience, and we're going to be talking about the neuron and why it is that neurons have an electric charge. This is also known as the resting potential, so we'll be building up our knowledge of why neurons have an electric charge from the very beginning by thinking about a cell in general. So in the beginning there was a cell, and the cell was just a spherical blob, okay? Nothing too complicated so far. Now, the fundamental purpose of having a cell is to define the difference between an inside and an outside. The fact that there's a lipid membrane allows the cell to control an environment and curate what is inside itself in a way that is separate from what's outside of itself. A bit of terminology here. Every time we talk about things that are inside the cell, we're going to be calling that intracellular, intra for in. And everything that's outside of the cell, including a lot of other stuff that's out there, we're going to be calling extracellular, extra being outside. So this cell is just like every other cell. It's, it could be a prokaryotic cell, it could be a liver cell, it could be an invertebrate cell, it could be any other kind of cell. But we're interested here in talking about neurons. And so a neuron, like some other cells, curate an internal environment that is different from the outside environment in the following particular way. It is able to maintain a concentration of charged particles and ions in a way that is different than what's on the outside of the cell. In particular here, in this part of the lecture, we're going to start really simple by focusing on only a couple of distinct ions. We're going to be talking about potassium, of which there's a lot inside the cell and less outside the cell. Sodium and chloride, of which there's a lot outside the cell and a lot less of it inside the cell. Now this difference is really important because it establishes a reason for these ions to try to go to where there's less of themselves if they could. But of course they can't because there's a cell membrane, and the cell membrane is impermeable to charged particles like cells, like ions. Now, there's a mnemonic here that I like to use to remember which one is which, right? Because I always get confused sometimes. The way I remember it is that cells evolved in the ocean, and the ocean is full of salt water, like sodium chloride. And so the extracellular environment is like the ocean, has lots of sodium and chloride. And then the inside of the cell is the opposite. It has less sodium and chloride, and it has a lot more potassium instead. So that's a little mnemonic that I like to use sometimes to remind of the ocean. Back to basics, right? Okay, so the resting potential we're going to explain in the following way. First, we're going to talk about the neurons have a steady resting potential of approximately -60 to -80 millivolts. Now, if you think about it, in terms of batteries, this is actually a really large voltage that we can measure. This is literally what you would measure if you were able to stick two ends of electrodes, one inside the cell and one outside the cell, and you read the little voltmeter. This is what you would read, -60 to -80 millivolts. Now, this is a large number because it's almost a tenth of a volt. And if you scaled it up to the size of something like a battery, like the one that I have in my clicker right here, that would be a 300,000 volt battery. So this is quite a bit of charge that's being maintained across the plasma membrane, across the lipid bilayer membrane that is the cellular membrane. Additionally, most neurons are highly permeable to potassium at rest. Now, I told you before that cell membranes are impermeable to ions, and that's true. Okay? The trick here is that the reason the cells do this is because they actually are then able to express proteins and then control which ions that they want to be permeable to at what times. So one observation we can make is that most neurons are actually pretty permeable to potassium at rest because they express specialized ion channels that are selective for potassium and allow potassium to pass in and out as they will. So basically it's like a door in the plasma membrane that's only open to potassium. Potassium can come in and out as they wish. It doesn't matter which way, the doors don't dictate which way they go, but they only allow potassium to pass through when the cell is at rest. Okay, we'll talk about active later. This is at rest. Now, all of this is true. These are just facts that are true that I'm telling you, but how does this even make sense? All right, we're going to be talking about in the next couple of slides about why it is that this is actually true and how this makes sense in the context of what we know about electricity and ion potentials. But a little bit of terminology first. So the membrane potential I'm going to be denoting as V sub m for voltage of the membrane. So anytime you see V sub m, that is the current potential of the membrane. Okay. There is a resting potential, which is just what the membrane potential is when nothing else particularly is happening. I'm calling that resting potential. So if you hear me say resting potential, that means the membrane potential at rest. That's the resting potential. Now, the resting potential tends to be a little bit negative, like we talked about. When it becomes more positive, I'm going to call that depolarizing because it is becoming closer to zero and less polar. If it becomes more negative, I'm going to be calling that hyperpolarizing because it's becoming more polarized because it's going to be farther away from a membrane potential of 0. So this is just a bit of terminology. I wanted to do this up front so that we can all be clear on what do these words mean. You can say a lot of different things in different ways, and so I think in neuroscience it's important to just get the lingo down up front. Okay, I'm going to be using these terms and I'm going to be using them interchangeably. So increasing potential, resting potential, membrane potential, depolarizing potential, these are all words that I might say in the course of talking about the foundations of how neurons are electrical cells. Okay, so let's think about the resting potential first. I told you that the cell at rest is very permeable to potassium. So let's make a vast simplification for now, and just let's ignore the other ions just for a little bit and just focus on potassium because it's much easier if we just think about one ion instead of a bunch of them. Okay, so I have a cell. And I have a bunch of potassium on the inside and a little less potassium on the outside. And I have a bunch of those little barrel-shaped things, which are my little symbol for potassium ions, places where potassium can go in and out if they want to. All right? Now, if you remember from chemistry, when you have a lot of something on one side and less of it on the other side, and doors for the thing to go through, what's going to happen is that the potassium is going to want to leave the cell, right? There's a concentration gradient, and there's a reason for the potassium to leave the cell so that it can balance the concentration on the inside and the outside of the cell. So the first thing that happens is as the potassium channels open, the potassium is going to want to leave the cell. All right? So there's a net outward movement of the potassium going outside of the cell. Now, that's fine if all you're talking about are regular solutes that are in solution, but we're talking about ionic species here because potassium is a positively charged ion. And when positively charged ions leave the cell, it actually makes the membrane voltage more negative. Right? So positive ions leaving the cell means there's more positive on the outside, which means that the inside ends up being more negative. So as the membrane potential becomes more negative, there is a balancing electrical force where the positively charged ions on the outside are drawn towards the more negative membrane potential on the inside. I'm going to say that again. There is a balance of electrical and concentration forces here, where the concentration force makes the potassium want to leave, but as it leaves, the membrane potential becomes more negative, which makes potassium want to come back in. These two forces are going to eventually balance. And the point at which it stops moving, when there's no net movement, when the electrical forces and the concentration forces are exactly balanced, is called the Nernst potential of potassium. This Nernst potential is super important because it tells us the point at which there is no more net movement of potassium in or outside of the cell, despite the fact that all the doors are still wide open. Now, of course, stochastically, because of thermodynamics, there's going to be a couple of ions that go in and out anyway, but the point here is that there's no more net movement, and therefore no more changes in the electrical potential, in the membrane potential of the cell, in my simple scenario where all I care about is potassium. Now, one more thing I'm going to point out, the Nernst potential here is a voltage. It is basically a target voltage. If this is all that's happening, if all we have is a cell and potassium and some ion channels that are permeable to potassium, then the Nernst potential is the voltage at which everything will come into an equilibrium because we have now balanced the electrical and the concentration forces. Everything's in balance and nothing's going to move any farther. So that is going to be the resting potential of this particular cell. That's what's going to happen here. So what's going to happen next is that we're going to be digging a little bit more into exactly how this Nernst potential is going to be computed. I told you intuitively that it's this place where electrical forces and concentration forces come to a balance. And so the way that we express this mathematically is by the following equation. If you wanted to actually compute what the Nernst potential of any particular ion is, here's the equation that you would use to do so. It's called the Nernst equation. And so the equilibrium potential of a particular species of ion, so here we're talking about K for potassium, is equivalently called the Nernst potential or the equilibrium potential. So here I'm denoting as E sub K, E for equilibrium. So this is the equilibrium potential of potassium. The factors that go into it are the following. We have a ratio of the inside versus the outside concentrations of potassium. So I'm literally going to take the outside concentration of the potassium and divide it by the inside concentration of potassium to get the ratio of in versus out of that particular ion. Notice here that technically, I don't actually need to know what are the concentrations on the inside and the outside. I just want to know their ratio, right? It doesn't matter if it's 10 moles versus 1 mole, 10 moles out versus 1 mole in, or 10 millimolars out versus 1 millimolar in. As long as the ratio is the same, this equation is going to give me the same Nernst potential, the same equilibrium potential. After we take the natural log of that, we're going to then multiply it by the following constant, where RT, R is the gas constant, and T is the temperature in Kelvin, and F is the Faraday constant. Now, you'll notice that these are things that are fairly standard in a standard physiological scenario. So if you are at room temperature or body temperature for a reasonably temperatured animal, R, T, and F are not going to be changing. So in fact, we can make the approximation that at room temperature, where you might be doing a reasonable experiment, sometimes we heat up. If you're doing a kind of a warmer animal, sometimes you heat it up. But at room temperature, RT over F is approximately 25 millivolts. And so we can use that almost as if it's a constant, because in most scenarios, it's not really changing all that much. Now it's RT over F, and then also to divide by Z. Z here is the valence of the ion. So for potassium, it is a positive ion of charge 1, so valence Z would be plus 1. Similarly for sodium, Z would be plus 1. For chlorine, because it's a negative 1 charged ion, valence would be negative 1 in this particular case. Okay, so Z does change depending on which ion you are looking at. So here we have a formula for computing the Nernst potential of any ion, as long as you know the ratio of its concentrations inside and outside, as well as the nature of the ion, what the valence of the ion is. In a slightly more complicated scenario, realistically, we actually have more than one type of ion. We have potassium and sodium, and this is an example of some plausible scenarios of what might be the concentrations in a normal animal cell in terms of the outside concentrations as well as the inside concentrations. If you crunch through the Nernst potential equation, you'll come upon the Nernst potential for each of these ions, and you'll realize that because of the opposition of their respective concentrations inside and outside of the cell, the Nernst potential for potassium is very negative. It's approximately negative 80 millivolts. Whereas the Nernst potential for potassium is quite positive. It's about plus 60 millivolts. Okay, so these are kind of opposites at this point. Now, all things being equal, you might think that sodium might want to rush into the cell, and potassium might want to rush out of the cell. So how does this happen? How does this even make sense? If sodium is always leaking in and potassium is always leaking out, why don't the concentrations just equilibrate? Why does it all come to exactly the same concentration? So the answer to this question has to do with the sodium-potassium pump. Now, this is the way that the cell tries to maintain equilibrium. Now, if you were a neuron, the fact that you have this resting potential that is not zero, where not everything is the same everywhere else, where there's a difference between your inside intracellular environment and the outside extracellular environment, is something you want to maintain because you're maintaining readiness. It is only at that maintained negative potential are you as a neuron ready to respond for the world. You wake up in the morning with a smile on your face, like, okay, bring it. I'm ready for whatever. This is how a cell, as a neuron, is ready, by maintaining that difference between the inside and the outside. And so it's really important for the cell to maintain a high concentration of potassium on the inside and to get all the sodium out. It needs to get out. All right? Now, this is such an important process that the pump, the protein that is responsible for maintaining this concentration gradient, is one of the primary reasons why your brain consumes so much energy. So your brain, despite weighing only approximately 2% of your body mass, consumes something like 20% of the calories in your body at rest. This is not true when you're running or doing something really physically rigorous, but if you're just sitting there and not really moving a lot, 20% of your calories that are being consumed every minute is going to your brain. So, and this pump is the reason why. This pump, unlike ion channels, which are just doors that are open, ions can come in, they can come out, whatever they want to do, it's totally cool. These pumps are significantly more bossy. They are actively using energy in the form of ATPs to pump ions against the concentration gradient, against the electrochemical gradient. In this particular case, we have a sodium-potassium pump that is super active in every single neuron, maintaining those ionic concentration potentials, where for every time it cycles, 3 sodium ions are pumped against the electrochemical gradient out of the cell, and then in the second stroke, 2 potassium ions are pumped inside. All right? And every time that happens, 1 ATP is hydrolyzed into ADP, and we have a net expenditure of energy and pumping of sodium and potassium ions respectively against the electrochemical gradient. This is so expensive that we can actually use this as a way of estimating how many ATP molecules does your neuron spend per second. Just doing this, right? Even if it's not doing much, it's ready to respond, right? Even when you're sleeping, you're ready to respond when there's a siren that goes off and you have to respond immediately or something like that. Even if you're just sitting there, you might be ready for somebody to tap on your shoulder and you have to turn around and respond to whatever that is. So your nervous system's job, part of its job is maintaining readiness. And so it expends an exorbitant amount of ATP molecules just being ready. So let's do a fun little order of magnitude computation about how many ATP molecules are spent by a single one of these neurons per second. Okay, so I'm going to do it the following way. I'm going to say that we're going to start with the following general prescription of a normal adult human for something like 2,000 calories per day-ish. We'll start there. It's close. Okay, it's same order of magnitude. It's not going to be 10 times smaller than this, and it's not going to be 10 times more than that for the vast majority of us. So we're going to start converting the calories per day humans, and we're going to convert it into 20% of those calories goes into running your brain. And we're going to convert calories into kilojoules into moles of ATP. We then multiply by Avogadro's number to get the number, that's not numbers. That's the number of neurons in your brain. So we're making the approximation here that you have approximately 86 billion neurons in your brain, okay? Then we multiply by Avogadro's number to get the number of ATPs in a mole. Everything cancels out so far. We then make the conversion of days, the number of seconds, to come upon the order of magnitude of about 1 billion ATP molecules are spent by every single one of your neurons every second of you just sitting there on the couch watching Netflix, not doing much. That is a lot of energy just to sit there being ready for something else to happen. So this is also, the Nernst potential, a really interesting way of being able to do some intuitive exercises in terms of what might be happening if something about the passive environment inside and outside of the cell starts to change. So I told you before that there's a lot of potassium on the inside and much less on the outside. What if we artificially and even temporarily increased the extracellular potassium concentration? So, for example, in an experimental context, this might be I have a neuron that I dissected out of a cockroach, and I put it in a dish, and I'm going to bathe into a solution that has a lot more potassium in the extracellular space than the physiological conditions actually are in the animal. Okay, so what if we just did that? What would happen? Okay, so if we reason through this, what's going to happen in a hand-wavy way is that the concentration force is going to be reduced, right? So there's more on the outside, which means that the concentration gradient, the forces pushing the potassium ions out, is going to be less. And so it's going to be reduced concentration forces. Therefore, potassium is going to start moving back into the cell because there's going to be a lower concentration force. The cell then depolarizes because we have positive ions moving into the cell, making the membrane potential more positive. And another way of saying the membrane potential being more positive is that it's depolarizing. And what's going to happen is that the electrical forces is then reduced, and we come to a new equilibrium that matches the concentration force. And that's what's going to happen as the membrane potential reaches the new Nernst potential, which we can compute quantitatively by taking the ratio of the inside and the outside potassium concentrations. So I'll leave that to you as an exercise to actually crunch the numbers. If you plug in these numbers, when you have 20 millimolars of potassium on the outside instead of 5 millimolars, how much does the Nernst potential actually change when we artificially increase extracellular potassium in this following way? So there's another interesting thought experiment that we can do. I told you already that all of these resting potentials are maintained by resting potassium ion channels that are open most of the time, even though the cell is not doing anything. Okay? Now these resting ion channels, even though they seem like they're not doing anything, they're actually kind of important, because it turns out things kind of go wrong if there's not enough resting ion potentials, because these are what's maintaining the resting potential of the cell. So, for example, you might reason that cells now have a reduced ability to maintain the resting potential, because the resting potential is predicated on the assumption that all of these potassium doors are open and the potassium come in and out as they want and reach an equilibrium. The other thing that's going to happen is the cells might become abnormally excitable because it's not able to return to this normal resting state like it normally would, right? If it got excited, it wouldn't really be able to come back as quickly as it really should be able to, as it normally would in a different cell if there were enough resting ion channels, okay? So this seems like a slightly silly scenario until you realize that there are actually goats that have this condition. This is what's called the fainting goats with congenital myotonia. And in this type of disease, these goats have not enough resting ion channels, which means that when they become excited, when they become startled, they tense up their muscles and contract, and they can't relax them. And because they can't relax their muscles and they're stuck in this tense state, they totally fall over, like that picture there. It's actually kind of adorable once you realize that they're only down for 10 or 15 seconds and they get back up as if nothing happened. They run around, they're totally happy again. This is a kind of a charming way of describing not enough resting ion channels in the congenital myotonia. But fainting goats is a much more lovely way of naming this condition. It's so lovely that it's actually the name of a local gelato shop that we have here in Seattle. They have 2 locations and they make really cool, they have really cool decor and they have good gelato. And so if you're in the neighborhood, go check them out. The Fainting Goat Gelato. I'm really not getting commission from them, not getting paid. I just like the gelato. So what's going to happen next is that we're going to then expand on this description of the Nernst equation. So far, we've mostly talked about potassium, and we've talked a little bit about sodium, but kind of separately. So we're going to start integrating this knowledge into a holistic view of the resting potential and trying to build up levels of realism by adding in things that are actually happening in the real cell without getting too complicated, at least not quite yet. So what's going to happen here is that, remember, we're caring about the ratio of the inside and the outside ion concentrations, and we also care about this constant here, okay? So this is a reminder, we just did this. And what we can do is think about the scenario of what if we had more types of ions? Okay, so what if instead of just having potassium ions and the relative concentrations in and out of potassium ions, what if we also had sodium ions? We could have other ions too, right? You can have calcium, you can have chlorine, you can have all kinds of ions that are in here. So let's just think about what would happen, okay? So intuitively, you might think that if you had more types of ions, the right thing to do here would be to, instead of thinking about the relative concentrations, the ratio of concentrations of just one type of ion, maybe you would want to be taking a weighted average of the concentrations of the ions. I'm going to say that one more time. Here, we have an equation that is the logarithm of just the ratio of this one type of ion. If you had more than one type of ion, you might want to take the weighted ratio of how much that ion is able to flow in and out of the cell. So for example, if we had sodium as well as potassium, you can have a similar ratio for sodium, right? Sodium out divided by sodium in. You have a different ratio. You have a ratio for potassium, ratio for sodium. As we saw earlier, the relative ratios of these ions inside and out of the cell can be quite different. And what you can then do is take an average of them. If the cell is equally permeable to both ions, if it is equally, all the doors are open, if it's equally permeable to sodium and potassium, you would just take the average of the two. If it is more permeable to potassium and less permeable to sodium, you might intuitively want to take a weighted average, where the ratio of the potassium is weighted more heavily than the ratio of the sodium. Right? So if it's like a 50-50, you just average them. If it's 70-30 or something like that, you might be like, okay, 70% important is potassium, 30% important is sodium. Okay? So that's the intuitive hand-wavy explanation of what to do if we have more types of ions. And in fact, that is exactly what we are going to do by writing down, instead of the Nernst equation, we're going to be writing down the Goldman-Hodgkin-Katz, otherwise known as the GHK equation. Now this equation, don't be afraid of notation. We're going to break this down because it's exactly what I said intuitively earlier. Instead of having just one ionic species like in the Nernst potential, we're going to be computing the equilibrium potential of the membrane, E for equilibrium, M for membrane, equilibrium potential of the membrane. RT over F is exactly the same as before, logarithm exactly the same as before, but now instead of having one ratio of outside versus in, we're going to be doing a weighted sum of all of the ions here. So let's break this down. So we've mostly been talking about cations so far. Let's just ignore the anions. We'll just talk about the cations, the positively charged ions. Okay? So the positively charged ions, for every single ion, we're going to do subscript J, right? J is every type of ion. So J equals 1 might be sodium, J equals 2 might be potassium. We're going to take this ratio of the outside versus the inside, and we're going to weigh it by the permeability to that ionic species. How permeable is it? And then we're going to add, this is the summation sign, we're going to add the weighted permeability by the ratio of the other ions. And as many as there are, we're just going to weigh them together. We're going to take a weighted sum of all of them. Cations are positively charged, anions are negatively charged. And so the net effect on the membrane potential of a positive ion moving in and a negative ion moving in is just going to be the opposite. And so for anions, we're going to do exactly the same thing, except it's going to be in divided by out instead of out divided by in, because they have opposite effects on the membrane potential. Now, all of this is kind of assuming that the cations and anions all have a valence of 1, right? So this is totally fine for the ionic species that we have over there, sodium, chloride, and potassium. If you had something that has a +2 charge, like calcium, magnesium, there's a slight modification we would have to make to this in order to get it to work, but you can kind of intuit that. I wanted to keep it not even more complicated in terms of mathematical notation. So this is what we would do to figure out what the equilibrium potential of a cell is if there were more than one ionic species with different permeability at rest to get to the resting potential. So next, we're going to do something really fun, which is think about what happens when things are moving around. We're going to take this intuition of ions moving in and out of the cell, kind of this hand-wavy intuitive explanation, and put it on a more firmer mathematical physical grounding by thinking about Ohm's law. Now, Ohm's law is something that you may have come across if you had done a little bit of study of physics, of electricity and magnetism, because it's one of these really foundational things that tie together a lot of different quantities in electricity and magnetism. In particular, it ties together voltage, which we talked about already, current, I, that's a new one, okay, and resistance, which is R. And we know that these three quantities are related by the equation V equals IR. The voltage is given as the product of current and resistance. We know this to be true. And this is going to be true in the cell in the same way that it is true in an electronic device or breadboard or something that you build out of wires with batteries that are macroscopic in a physics lab or in electrical engineering lab. So Ohm's law, we're going to break this apart, and I'm going to break down what Ohm's law means for us in the context of ions moving in and out of a neuron in terms of the passive potential and what happens to this when it happens to the cell when it's slightly perturbed from rest. So we're going to rearrange this equation a tiny bit, right? We're just going to take I, going to leave I on one side and bring R to the other side. And so instead of V equals IR, we have I equals V divided by R. Next, I'm going to just do a tiny bit of sleight of hand, where instead of having resistance, I'm going to talk about the inverse of resistance. I'm going to talk about 1 over resistance, also known as conductance. I'm going to denote this by the letter G. So anytime you see small letter G, that actually means conductance. Okay? Now, conductance, in the context of ions moving in and out of the cell, is a little bit different than what conductance means when you're studying in terms of wires. So for example, we know that a copper wire has more conductance than a tin wire. It's a better conductor of electricity. It's also a better conductor of heat. But anyway, so conductance in the context of Ohm's law for wires just means how easily electricity passes through that material. For something like a normal piece of plastic, conductance would be very low. It's basically an insulator. It doesn't really conduct electricity at all, right? Okay, so that's what it means in terms of wires and electronics. But here, instead of talking about electricity passing through a wire, we're talking about ions going in and out of the cell. Because the only way for that ion to go in and out of the cell is through that membrane pore, that protein that is an ion channel. Conductance in this particular case is directly proportional to the number of ion channels through which that ion can pass. More ion channels, higher conductance. That's really all it is here, right? So that's the interpretation of Ohm's law, where V is something proportional to the membrane voltage. I is current, and again, current here, instead of being flow of electrons in a wire or electronic device, we're talking about flow of ions in and out of the cell. So I here is actually flow of ions in and out of the cell. So the flow of ions in and out of the cell is going to be proportional product of the voltage, some kind of voltage driving force, and the conductance, the number of open ion channels through which that ion can pass. That's what we're expressing here in this version of the Ohm's law, interpreted in the lens of neuroscience and the electrical properties of the neuron. We're going to take this equation and we're going to just work with it for a little bit further. We're going to move some stuff around and see how far we can get as we do. This is what happens. Okay, so we have this equation. And what's going to happen next is we are going to consider what happens in the case of the potassium ion. So here I'm going to write I sub k because it's the current that's associated with the movement of potassium ions in particular. Again, we're just talking about potassium for now. There's other ions being moved around, so there's other currents. Right now, we're just talking about potassium. So the movement of potassium in and out of the cell is associated with a current, and the magnitude of that current is going to be proportional to the following term, G times V, okay? G, like I said before, is quite literally the number of ions that the ion channels that are open. This V term here takes a little bit more explanation, so I'm gonna break this down for a little bit. V is not actually just the membrane voltage. V is kind of like the electrical voltage force that's associated, how much something wants to go to somewhere else. So here, it's the difference between the membrane voltage, wherever it is, whatever the current membrane voltage is, and the difference between that and the equilibrium Nernst potential of that ion. Okay? And G sub k is the conductance of potassium, which, since the only way potassium can go anywhere in and out of the cell is through ion channels, it is also the number of ion channels that are open. Okay, so those are all the ingredients of this equation. Let's think about that for a second to make sure this makes sense to us. Okay? So the flow of ions tells me it's a net flow of ions, right? Net flow of ions is going to be proportional to V sub n minus E sub k times G sub k. So let's see if this makes sense, if this jives with our intuition we think should be happening here. G sub k is the number of ion channels that are open. If G sub k were 0, like there's just no ion channels open, then this quantity is 0. Doesn't matter what the other stuff is. I is going to be 0 because there's no ion channels open. Does that make sense? Okay, it does, right? If there's no ion channels open, nobody's going anywhere, so the flow of ions is going to be 0. Okay, so far so good, right? Now let's look at this other term here. What if V sub m, the membrane voltage, is exactly the same as the Nernst potential of potassium? So we're already there. We're already at the target equilibrium potential. This quantity, right, this subtraction will give us also 0, which means that flow of ions is once again 0. This also makes sense because, as we talked about earlier, the Nernst potential is kind of the target equilibrium potential, right? And if you're already at the equilibrium potential where the electrical chemical forces are already at balance, then there should be no net flow of ions anywhere. So once again, this jives with what we learned before, where we can reason through how this equation makes sense in the context of what we think we know about the physiology of the cell, as well as what it means for the cell to be at an equilibrium Nernst potential. Okay, so we're going to take this equation here and make the following observation. It turns out that this equation is exactly how we would express everything we just talked about in terms of an RC circuit that you would actually build in either a physics or electrical engineering lab on a breadboard. This is known as the RC circuit because it's super well studied. It has a resistor and a capacitor. So this exact equation that we just built up is the same equation that describes our resting potential of the cell with potassium, and it's the same equation that describes this circuit right here. Exact same thing. They're equivalent. I'm going to say that a couple of different ways. So if you were trying to study the resting potential of the neuron from the perspective of potassium, you may as well think about it with the analogy that is acting exactly like that circuit that we can draw over there, where extracellular is on the outside and intracellular is on the inside. The voltage is the voltage between the inside and the outside. We can totally measure that if they were macroscopic into something like the little circuit that you built on a breadboard. And it has two parallel paths, where the right-hand path is the capacitance, and that corresponds to the capacitance of the membrane, C sub m, capacitance of the membrane, where there's negative charge on the inside and positive charge on the outside, therefore V sub m is going to be negative because it's negatively charged overall. And the other parallel path is how the ions can pass through the inside and the outside of the cell, where there's going to be a conductance. That's actually a resistor. There's a resistor whose magnitude is 1 over G. Remember, G is 1 over R, so R is also 1 over G, right? That corresponds, G corresponds to the number of ion channels that are open. And then we have an equilibrium potential there, corresponding to the corresponding potassium, E sub K. So this and the picture I drew earlier of the cell and that RC circuit are just different ways of thinking about exactly the same system. And we can go back and forth, but using each description as necessary to reason through the scientific findings that we're trying to explain. And so I'm putting this up there not to be extra confusing, and not because you need to be able to understand every single way of doing it, but rather because in neuroscience, a lot of what we're doing is drawing analogies with other disciplines, other cognitive and physical and computational disciplines, because it's a really good way for us to draw connections between how we think the brain works and what we think we understand about how other systems work. And so it's not important for every single person to be equally comfortable with every single description of it. Maybe you really like the equations, but you don't like the diagram. That's totally fine. Maybe you really like the breadboard diagram because you understand that from another class you took, but you don't really understand the Nernst potential equations and all the stuff about concentration grading because you just don't know chemistry that well. That's totally fine too. Maybe you just like the mathematical description of it because you understand mathematics and you don't really care for the electrical engineering diagrams. That's totally fine. I'm going to show you a lot of different ways to understand the same thing in order to draw connections, and if at least one of those connections connects with you, then I'm pretty happy and I feel like I've done my job here. So that's the RC equivalent of the flow of ions, and we can once again use the same equations to get to a more quantitative understanding of what happens if we increase extracellular potassium. So earlier on, in the previous section of the lecture, we talked about what happens if we increase extracellular potassium in sort of a hand-wavy way by thinking about the direction in which things would move. Okay? Now, armed with this equation, I want you to actually work through the numbers and quantitatively think about what might happen if we increase extracellular potassium. What would happen to each of these terms, right? So we're trying to get to what happens to the flow of ions. We know something about membrane voltage. Does that change if we change extracellular potassium? What about the equilibrium potential? Does that change if we change extracellular potassium? Does it change the number of ion channels that are open in this scenario? Work through each of those exercises in your head and try to come to an understanding of a quantitative understanding of what happens if we increase extracellular potassium and by how much. What if it actually exceeds the intracellular potassium at some point? What happens then? This equation gives us the ability to make those predictions not only hand-wavy and intuitive, but also quantitative as well. And that's where the power is of these different types of description. So when we have more ions, we also have more channels, and we can make an equivalent description where we can expand on our RC circuit by putting in more parallel pathways, one each for each of the ionic species. Now, they're independent parallel pathways in this model because they're actually independent, right? I told you earlier that potassium channels are just permeable to potassium. If your sodium can't go through, use your own door, okay? And so if you do it that way, then what we can see is that we can then just write down exactly the same equations like we write down for potassium, and we can write them down one each for each ion because they're independent. They actually have nothing to do with each other except they're tied together by the current membrane voltage. So we can do more ions by adding more channels. If there's different types of channels, we can write more equations for how they affect the conductance. If there's different ions, if there's more ions than the three that we've done here, we can just add more equations and to have a more complete description of what's happening relating, using Ohm's law, current, voltage, and conductance. So I told you already that the reason we can write these equations independently is because they have these completely independent parallel pathways where the sodium channels are permeable only to sodium. You can't come through if you're potassium. Potassium has its own channel and it's not permeable to sodium. So molecularly, from a protein level, how is it that these ion channels are so choosy? How do they do that? That seems really difficult, right? Because I can kind of see how you might get, you can have a negative ion can't pass through a door for positive ion, but sodium, potassium, they're both positive ions of charge +1. How is an ion channel as a protein machine possibly able to be so choosy? So this is a really fun story, and I'm going to share it with you in at least conceptual terms. So ion channels are awesome proteins. I mean, they kind of all are awesome proteins. Okay, but anyway, ion channels are awesome proteins. They're tetramers, which means that they have 4 subunits. And when they're assembled on the extracellular membrane, they form this helix where there's a hole in the middle. And the hole in the middle is where the ion is actually going to pass through. So as you remember from chemistry, ions are positively charged, and so they're stable in the aqueous solution. They're stable in the water solution on the outside, and they're stable with the watery solution on the inside. They can't pass through the lipid bilayer because it's hydrophobic. And that's the job of the ion channels, to form a friendly environment through which the ions can actually pass, where it's advantageous for it to pass through, to go from the inside or the outside to the inside. Either way, okay? These ion channels are not selective for direction. They're selective for ionic species. So to dig into it a little bit further, if you look in the actual pore area, zoom in to just the part of the protein through which the ions will actually go, what you'll see is that those 4 protein helices have carbonyl groups in the functional groups of the amino acids that are lining the edges of that pore, okay? And those carbonyl groups mimic the interaction of potassium ions with water. And so that's how it makes it possible for potassium to pass through the otherwise hydrophobic environment of the lipid membrane. The pore size of potassium is designed so that it perfectly fits and stabilizes the ionic structure of potassium, but not the smaller sodium ions. So even though they have the same charge, the sodium ions are actually too small to go through this pore, because when it goes through the pore, it doesn't interact with all of the 4 sides of the helices equally. So it's actually not happy going through the pore. you can kind of imagine the other way being a little bit easier, right? So if you had a sodium channel that stabilized the sodium ions, the potassium ions are just a little too big to go through. So that kind of makes sense that you might be able to build something like that. But it's interesting thinking about how the pore for the sodium channels, for the potassium ions, are actually too big, and the smaller positively charged ions are unable to go through. So that's the mechanism that ion channels are able to choose. I'll clarify once again that these are channels. They're selective for ions, but not for direction. When they're open, they're just open. A potassiumic ion can equally well use the same ionic channel to go from the outside to in or from the inside to out. The directionality is entirely determined by the electrochemical gradient, just like we talked about in the Nernst potential. The ion channel simply is the conductance, right? It's the equivalent of the conductance term. It's open, which means that flow can happen. It does not determine the direction of the flow. Unlike the potassium-sodium ATPase pump we were talking about earlier, which, because it's pumping everything against the electrochemical gradient, has a very distinct directionality. It only pumps in one direction and it does not pump the other way. Okay, so as a summary of this section of the lecture, here are the key concepts that I talked about to understand the resting potential of the neuron. The first topic is that neurons have an electrical charge between the inside and the outside in a way that is similar to but different from electrical charge works in physics or electrical engineering. Here, the charge is because of a difference in the concentration of small molecules that happen to be charged ions. We talked a lot about sodium and potassium as examples where there's differences in concentration, and their relative concentration is the reason why there is a net electrical charge between the inside and the outside of a neuron. Next, we talked about how this membrane potential is called the membrane potential. This electrical charge is called the membrane potential, and every time you see a big V, that means voltage, and sub m, little m, means membrane. So V sub n, every time you see that, it means membrane potential. So the balance of electrical and chemical forces determines the resting potential of the neuron, quantitatively speaking. Where those two forces, they're always going to be pushing and pulling against each other. Sometimes one is pushing ions out, the other one is pushing ions in, but they always balance out. And the point at which they balance is the resting potential of the neuron for all of the net species. To determine the point at which they balance, we use the Nernst equation, or the slightly more complicated Goldman-Hodgkin-Katz equation, which is related to the Nernst equation, and it allows us to compute quantitatively in terms of units of voltage what is the equilibrium potential either for one type of ion or for a mixture of ions that are of different permeability. We then talked about Ohm's law as a way of allowing us to determine the flow of ions in and out of the cell, right? We can intuit what happens from the Nernst equation by saying things should be coming in or coming out. Ohm's law allows us to determine how much the ions are going in and out of the cell. And then the resting potential, because it's so important for the readiness of the neuron to respond to whatever it is that we wanted to respond to, actively pump sodium-potassium channels against the respective concentration gradients to maintain what is known as the resting potential. And this is one of the primary reasons your brain is such a metabolically active and expensive organ to maintain. These are the key concepts, and I hope to see you for the next lecture where we're going to talk about something significantly more exciting, which is what happens when things are not just resting.